So, welcome all. My name is Malin Melin. It's nice to see so many of you here at this seminar on comprehensive genomic profiling and routine cancer care, where we will hear about the experiences here in Uppsala. We have three speakers today, Johan Botling, Gilbert Lauter, and Jonas Almlöf, who will tell us about different aspects of developing and using broad cancer panels for diagnostics. Before we start, I would like to ask you to keep your microphones muted. We will have time for questions and discussions at the end, so please write your questions using the chat function, and we will get back to them. This presentation will be recorded, but we will stop the recording before the Q&A session. So I believe that people have joined, so with that I will leave the word to Johan.
Thank you very much, Malin, for the kind introduction. So we will share this presentation, the three of us, and I will start with a short background and some thoughts about what comprehensive genomic profiling really is and the impact of the potential impact of these platforms. Also, we would like to point out that a lot of this work has been done in the GMS solid tumor working group and in different Genomic Medicine Sweden at different nodes, but we will focus on the Uppsala experiences here. So there is a lot more, of course, to hear from other nodes in the future around this project, of course. But I will start with some history. I think when you try to project what will happen in the next year, it's always useful to look back and see how did we get where we are today. And during my time as medical director of our molecular pathology lab, there has been really three big technology leaps, which I think are interesting to consider. The first one in 2010, we moved from Sanger sequencing to small singleplex assays, and this was really pushed by a need for higher sensitivity that we couldn't get from Sanger sequencing. We have worked a lot all over Sweden with pyrosequencing and real-time PCR assays, mostly. Then in 2015, we gradually moved to small NGS panels, targeted panels, amplicon-based, and this was pushed by a need for a wider target range, at least in selected tumors, and eventually also fusion gene profiling in a more multiplex fashion. So these are the platforms that really are the backbone of the clinical molecular pathology labs today. And now, recently, the next leap has been to move to broader comprehensive genomic profiling panels, such as Foundation 1, TSO 500, and now the GMS panel. And this move is motivated, of course, by all these words that we talk about every day: precision medicine, pan-cancer, all targets, mutation signatures. And also from the pathology side, we would like to point out that these panels also are extremely useful in tumor subclassification for routine diagnostic pathology. I will get back to this issue because I think it has been underestimated and underdeveloped in these kinds of panels so far. Also, the history tells us that these leaps occur maybe every 5 years or so. I think this is quite interesting because it shows that moving to a new platform is a lot of work, and you need to be very sure about assured that these new platforms must work in real life, maybe for 5 years or so, and should optimally be flexible and allow for some adaptation to needs that arise in the coming years. So what will happen with the broad CGP panels? Well, looking back 5 years, this is how the small panels were introduced in Swedish healthcare. You see from 2015 to 19, pretty much over a period of 5 years, the volume increased a lot, and also the domination of these panels as the standard tool also happened during quite a long time period. And I think looking ahead, I think it will, my guess is that it will be the same over the coming 5 years, that the broad panels will take over gradually over quite a long period of time, while we keep a lot of the assays we run currently side by side together with the developing CGP panels. The history also tells us that we need an RNA dimension to new platforms. So this graph shows the introduction in light gray of RNA-based small panel sequencing in Sweden, and this has mainly been driven by lung cancer, the need to assess multiple fusion genes in lung cancer. So this is where we are today, with small diagnostic NGS panels implemented all across Sweden. Different panels. This was very important for the formation of the solid tumor working group, because we had before GMS and clinical genomics, this area worked a lot together, helping each other out with these different platforms. Also, I would like to highlight, I like this VM Guld picture, because as far as I know, we are the only country in the world that has national coverage for patients with NGS panels. That is quite unique and something we should be very proud of. And this is also well recognized by the pharma industry. Okay, so why moving to comprehensive genomic profiling? I will introduce this concept by comparing to the alternatives. As you see to the left here, the small diagnostic NGS panels have been very successful thanks to good adaptation to small biopsies and cytology, a very high sensitivity and specificity thanks to very deep sequencing, and good adaptation to formalin fixation in routine pathology. Of course, the setback is that these panels work fine for selected cancers, and importantly, they are limited to point mutations and indels only, and not other aberrations. And you need a separate panel to look for the RNA dimension, that is the fusion genes. In parallel over the years, the research tools have been running with global assays, whole genome, whole exome, RNA-seq. Of course, they are fantastic. They cover all the genes you want and all types of aberrations. However, the setbacks here is that in principle, you need fresh tumor samples, which are seldom available in clinical routine. They are more expensive, laborious, and create enormous data sets. and actually have limited sensitivity and specificity, at least as they are run in research mode. In principle, a sensitivity comparable to Sanger sequencing. And as you remember, we abandoned Sanger sequencing 10 years ago because of sensitivity issues. So by combining some of the wide range of research tools with the clinical adaptation from the small panels, you get these comprehensive genomic profiling panels, usually around 500 genes on the DNA level, with different RNA capture modules on the side. So doing this, you can actually combine a very high diagnostic precision with a wide range of treatment targets and diagnostic targets, at least considering the treatment landscape today. And I think this concept has been very successful and proven in real life by a very big use in, for example, by Memorial Sloan Kettering in their publications, Foundation One as a commercial panel, and the last years also the adoption of Illumina's TSO 500 across the world. Okay, and I think that we are in the right track. This is the strategic document from Genomics England that was published last month, and they are now also focusing on these large panels for solid cancer and blood cancers in a combo, of course, with parallel techniques such as histopathology, immunohistochemistry, and imaging, developing imaging tools. Okay, another important aspect of new platforms is that you need to check with the clinicians what they want. And this is quite interesting, I think, because there has been a very fierce and heated debate if broad sequencing is needed at all in clinical routine. And there have been two camps among oncologists. Some oncologists think that this is just for fun. You never find something useful that you can use in routine oncology practice. Some other oncologists, they think this is, the future, that sequencing can solve everything. The truth is, of course, somewhere in between those standpoints. So we have worked with the Illumina TSO 500 platform for a while now. We have been running it in clinical routine for 3 years, not a big volume, around 250 cases. And when I look at our results now, from a perspective case by case, did we find something that would be useful for a Swedish oncologist? Yes or no? Or did we find something that is useful for a Swedish pathologist making a correct diagnosis or a tumor subclassification? And when combining these hit rates, in my opinion, we reach a hit rate at around 32%, where we find something useful, which is quite high actually, and for me motivates implementation of these panels now in clinical routine. I will just give you some short examples of what we have found in different cases that illustrates some different points. This was a young woman with a strange or a tumor of unknown origin. Sequencing here revealed one of the few NTRK fusions that we have detected. And of course, this is a very strong treatment target that could be used for therapy. Here is another case with a strange tumor in the pelvis. It was unclear what to test for, so we did a broad panel. We found microsatellite instability, high TMB, and a mutation in BRAF. And of course, these represent two different treatment options with immune therapy or possibly with BRAF-MEK, combined BRAF-MEK inhibition. So a treatment impact here, together, I think, with the diagnostic impact as MSI-related carcinomas in this area should represent a colorectal cancer or an endometrial carcinoma. This was a young boy with a strange sarcoma in the abdomen. The fusion gene that we found is actually diagnostic of a specific entity in sarcoma that I never heard about before, and also a treatment target. Same thing here, a strange melanocytic lesion, a therapeutic implication with a ROS1 fusion, also diagnostic implications, as these kinds of fusions are common in so-called spitzoid melanocytic neoplasms. Here, an unclear case where the tumor origin wasn't known. Was this a lung or a kidney cancer? Well, the mutations we found were typical kidney cancer mutations, so diagnostic impact here. Okay, so these are some examples illustrating sort of the clinical impact of broad panels. And finally, my summary slide is that, of course, the word comprehensive profiling for me means that we can do both diagnostics and treatment targets. We cover many cancer types. It's both DNA and RNA based. It covers many types of aberrations and can replace many of the current assays we actually run in routine every week today. So with that, I will hand over to Gilbert to talk about the GMS panel.
Yeah. Yes. Okay. Yeah. Hello, everybody. So I would like to present our work about the validation of the GMS 560 panel, a little bit how we worked and how the panel performed, and also some experience from the clinic. But first, some more background information about the actual panel. So the design strategy got certainly input and guidance from existing broad panels like the MSK Impact Foundation one and TSO 500, but also from Johan Lindberg's panel in Stockholm. And the aim was to certainly include possible targets for existing and upcoming therapies. There has also been input from the neuropathology on choosing diagnostic targets that have been included. Then there is a set of fusion genes that can be detected both on the DNA, but also on the RNA level. And of course, they are also included common oncogenes that explain the tumor biology behind different cancer types. More technically, the total size of the panel is around 2.5 megabases now, and includes, as the name suggests, 560 genes with exon coverage. There's also included a structural backbone. and a focused design for detection of loss of heterozygosity in 19 genes. Furthermore, there is a pharmacogenomics module to detect or to assess the genotypes of genes that are important or influence drug response. Also, there is like 44 ID SNPs that can be used to avoid mixing of patient samples. And then, as I said, there's also add-on. In addition to DNA, there's a separate RNA panel to detect 44 fusion genes. So all these components make it possible that we can make the switch from these more focused, smaller gene panels to the broad gene panels, so that where we can look at many genes and also many more different types of aberrations. So we can assess single nucleotide variants, CNVs in those, but also structural variants like gene fusions. And then we have the more complex biomarkers, HRD, homologous repair deficiency, MSI, and also tumor mutational burden. Okay. To the validation, well, the aim was to validate a broad gene panel nationwide and to implement it across Sweden. So for this purpose, all 7 centers joined, and the workload was split into different work packages and then assigned to 3 different working groups that worked closely together, to solve more practical problems. Then the workflow itself, we actually invested quite a lot of time to agree upon a consensus protocol. So we adjusted incubation times, temperatures, cycles, concentrations, and so on to get a consensus protocol that everyone tries to follow as close as possible. The whole library prep is a combination of the kappa library prep and twist target enrichment. The samples in the validation the majority are definitely FFPE material, but we also tested some fresh frozen and blood is also included. Then there was a national pipeline developed now, but this you will hear more about in the next part by Jonas. Okay, the validation itself, the setup was organized with the help of worksheets from the College of American Pathologists. So this helped us quite a lot to organize the work. In total, every center contributed with samples. In total, we have now included 200 known patient tumor samples, together with 50 matched patient normal samples. And we, of course, also run a whole bunch of commercially available controls, like the known HubMap control and 2 different types of OncoSpan controls. Also would like to add that on top of this, there are samples tested in the ongoing lung and breast cancer pilots. In numbers, in total, we have now 288 SNVs and 100 indes ranging from short to large. There are 73 CNVs included and 9 samples with fusions, but this is only on the DNA validation part. So I will talk a little bit more also about the RNA fusion validation. And then regarding the biomarkers, we have 45 samples to assess the HRD score, 72 for MSI, and 50 TMB samples. So quite a substantial cohort. And after sequencing all these samples and 31.7 billion reads later, I can present some results. But actually, I only focus here on the Uppsala part. So we ran all our samples on the NextSec, and The samples had a range of all different DNA qualities. And looking at the sequence coverage, so when we look at the median sequence coverage of all samples, we see that they 98.8% lie above 150x, which is the common acceptance threshold in TSU 500. Then if you look at the median coverage in clinical relevant positions, this is plotted on top of here in orange, then you actually get a higher median coverage of 300x in 96% of the samples. And looking at position-wise, you get in 92.5% coverage over 300 in 90% of the position in 90% of the samples. So overall, we get using even next-seq sequencing, we get a success, a very good coverage. Switching to the validation of SNVs. So in our cohort, there were 198 SNVs included, and out of these, 195 were called. So there were 3 missing that were not called, but this is because they are around 1%, so they are filtered out. But when we look in the raw data, they are still there. Also, of the 41 inlays that ranged from 1 base pair to 136 base pair, all were called. And looking at the correlation plots, you see that you get very nice correlation also indicated here by the R-square score. Okay, regarding TMB, so in our cohort we had 50 TMB, as known TMB samples, and could compare them with data from previously run Foundation 1 and TSU 500. We get a also nice correlation here. We get a somehow lower sensitivity when compared to Foundation 1, which is the golden standard. But this is because there are like 3 samples that lie just above the threshold that determines if it's TMB high or TMB low in Foundation 1, but below in GMS 560. Okay. Looking at the copy number variance, or more specifically at copy number gains here. Now you see in this table diagram that it's also a good correlation. In the next release, there will be improvement of detection of 1p and 90q and deletions based on a boosted backbone design. Regarding microsatellite instability, so there are 4,000 MSI sites in the design to determine the MSI status. So this is data from our smaller local validation of MSI, where we had samples of known MSI high and MSI low. And you can clearly see that the 2 populations are distinctively separated. We also include here the rest of our cohort, which is supposed to be MSS, and this is also true, except there was 1 sample that was an outlier, but there is a rational explanation, as this sample had a mutation in a DNA repair gene pole and was also TMB high. An upcoming module of the GMS 560 is the homologous recombination deficiency score. So this development is led basically by Lund, but also with input and data support from other sides. The score is a more complex one, or composite of 3 different genome stability scores. So far, there have been 45 samples sent to Myriad to get a known HRD score. And the samples really cover the whole spectrum of scores from very low to very high. And you see also that the correlation is rather good across the whole spectrum. These features will be hopefully soon available. And there's currently some more developmental work done looking at samples of bad DNA quality. Okay, another module work in progress is the pharmacogenomics module, which is also a joint venture with the Mia Vadelius and GMS Pharmacogenetics. So the rationale behind is to help or assist clinicals to understand how certain genotypes influence drug response and drug dosage, so which is relevant to determine the right dosage and to avoid any unnecessary toxicity. So currently the genotypes assessed focus on 3 genes, and the idea is that the clinician then can decide if they want to have this information and to get it as a kind of add-on feature to the standard GMS 560 analysis. And then there will be also a small report from a pathologist added to the clinical report. Now jumping to the RNA fusion part. So this is a separate working group and a separate validation also. The design of the RNA panel is based on junction capture, which basically means that you tile your probes at the end of putative junction positions. In this way, there are now 44 genes covered for the detection of fusions. And the workflow is basically the same. So it's also kappa twist, which makes it easy to incorporate it into the workflow with the DNA panel. And of course, you have to add a cDNA synthesis step specific to the RNA. The material tested in the Uppsala part, we have now looked at 20 different fusions, and there will be 30 more tested in the national validation. And of course, there are also commercial controls included. The fusions are, or the calling of fusions, there are 3 different callers, and at least 2 have to be positive to call it detected. So far, it has worked really well. So all fusions that were detectable have also been found. Just very briefly on clinical implementation. So we have now switched to GMS 560, like 3 weeks ago. And the transition was rather smoothly coming from a broad panel switching to another one. We have included both parts, so it's a GMS DNA part and the RNA part with a broader fusion panel. It also includes improved filtering, which means that we have less variants that we have to look at individually, but we still do this, of course. And then we have QCIs or QIAGEN's software to help us with the classification. In the next step, we have then a discussion with the pathologist, which writes a statement and goes through the data. This is just a snapshot how such a clinical report can look like. So here you have this summary statement and information about the findings of the biomarkers, and then the different variants are listed according to relevance. So if you have a treatable one, this will come up first. I would also like to point out that there are pilot projects ongoing to test GMS 560 in the clinical routine, both for breast and lung cancer right now. And just as a teaser, I would like to mention that the GMS 560 is also available for research projects via the SciLife Clinical Genomics platform. And with this, I would like to hand over to Jonas.
Yes, so my name is Jonas Almlöf. I'm a bioinformatician in Clinical Genomics Uppsala. I'm going to present the bioinformatics part of the GMS 560 project. So when we started this project, we realized that this will not be the only pipeline we will use. So we developed a framework for pipelines that we call hydrogenetics, which is available on GitHub. In this hydrogenetics, we have modules where we collect all the tools that perform the same task. For example, in the SNV-Indel module, you collect all the variant callers. And then when you make a new pipeline, you can just import these modules, which makes the job much easier. And hydrogenetics is based on SnakeMake, which is just a way to you make rules where you specify your input files and your output files. And then in the pipeline, you just specify your final output files, your result files. And then SnakeMake will find the correct rules to generate these final output files for you automatically. So it handles all the jobs, it sends them to the cluster, so you don't need to worry about these kinds of stuff. And so using this pipeline framework, the hydrogenetics, we have a number of pipelines under development, foremost and the GMS 560 solid pipeline with both DNA and RNA. We also have a hematology pipeline, a pipeline for hereditary cancer, whole genome cancer with both tumor normal and tumor only, and the pipelines for rare diseases. So except that it's easier to develop this pipeline with this framework, it also increases the quality of the pipeline. For example, we're using GitHub, and we enforce that you need to have someone else reviewing your code when you're merging your code. And also when you merge new code into the pipeline, it's automatically tested. For example, using dry runs or on small data sets. All the modules are also released with version numbers, and then at the release, we have extra controls on the modules, and to make it reproducible and portable, we use containers for all tools, which we have on our own Docker Hub. So we have full control of this. And the new feature also in GMS 560 is that when we make a new release of this one, we have an automatic comparison with the results of the previous release. We run it on a number of test samples. So we can see if the changes we have introduced produce the changes we want in the results. We also have documents on how to code standards, and documentation on the modules, even though this could be improved. So the GMS 560 pipeline has been, the development lead has been done in Uppsala, but we have many contributors throughout the country in Umeå, Göteborg, Linköping, and Lund on the hospitals there. And we also have users in Umeå, Göteborg, Linköping, and Örebro that has been running this pipeline, mostly so far for testing and validation. So this is an overview of the pipeline, the DNA pipeline. You start with FASTQ files, you trim them using the pre-alignment module, you align, you make alignment, and then you call variants, small variants, copy number variants, and fusions. And then we also calculate biomarkers like TMB, MSI, and HRD. Variants also annotated and filtered. And finally, we also get a quality control report. So if you start with the small variants, use two different callers, Mutec2 and Vardig, that we merge the variants from. We annotate them using web, but we also add annotations about artifacts that we get from a panel of normal samples that we have. Then we get a lot of variants to get this down to manageable level. We filter. In the present configuration, we keep only variants in exons, and we also filter out everything under 1% allele frequency. We take away germ SNPs and artifacts found in more than 3 of the normals. And we demand that we should have more than 20 observations in the reads. Finally, we add also what we call double variants here, and that is when you have multiple variants in the same codon, they are sort of combined and annotated and added to the VCF. And this is because a combined variant can have a different effect on the amino acid than the variants by themselves. And as Gilbert said before, we had really good results. We find all the variants if you go above 5% allele frequency, and also good performance under 5%. For copy number variants, we also use 2 callers, CNV-KIT and GTK CNV. Here we also use a panel of normal to filter out normal variation. We make a report where we only report on clinically relevant genes, which is about 20 genes for the amplifications and 20 genes for the deletions. For the amplification, we need more than 6 copies, and for deletions, less than 1.4 copies. And they should not be observed in more than 50% of the normals. So you get then a sort of list of CNVs, but you also get a HTML report, interactive one. Looks something like this, where you in the upper figure have the actual data over the entire genome. Then you can click on different chromosomes and get zoomed in views, also interactively zoom in if you have any special region of interest. And you can also click in the table to get to the variants directly. And this, I think, is crucial because every copy number variant you need to sort of confirm visually to be sure that they are true. And for amplification, this works really well, but as Gilbert said, for deletions, it's still under sort of development. For fusions in the DNA, we use GeneFuse. We also filter away sort of the noise if you have 2 fuse supporting reads. And this works really well if you have both the fusions, both the fusions in the fusion pair sort of included in the analysis. But it would take too long time to have all genes included. So for those fusions where you don't have both pairs included, then it won't find it. So, and it also reports very few false positives. So it works okay, but it's still sort of can't find everything. Then we go on to biomarkers. We have MSI, where we use MSI Sensor Pro, with then very good separation between MSI and MSS. For TMB, we have an in-house script that basically have a strict filtering of the SNVs. There we count the number of non-synonymous SNVs in this file and just divide by the size of the panel, showing good correlation with TSO 500 and Foundation 1. For HRD, it's, as Gilbert said, still under development, but it shows very promising results. So we use then the output from seeing the kit and puts that into a SCAR HRD, and you get a HRD score, which has good correlation with the mirrored scores. And finally, you get a QC report out from MultiQC, HTML report, looking something like this, at least the first table here, where we collect data from programs like FastQC, Picard, and SAMTools, and present that in sort of a way that you can quite fast see if everything looks okay. Then after the DNA pipeline, you can also run the pharmacogenetics if you like. It's not converted to the hydrogenetics framework yet, but it's done by Massi in Linköping. And I should also say that the original pipeline has been developed by Johan Botling here in Uppsala. For the RNA, I think I've gotten the wrong number here of the number of genes. I think it should then be GMS 44. But it's similar to DNA, that you start with the FASTQ files, you do trimming, alignment, and infusion calling, and of course, again, filtering. You also get some ID SNPs that you can use for contamination or sample mix-up, and the QC report. So the most important part here is the fusion, and we use 3 different callers. We use Ariba, which is fast but not so sensitive. We use Star Fusion. This has sort of medium computational speed and a good trade-off between false negatives and false positives. We have Fusion Catcher, which is slow and sensitive, but also generates quite a lot of false positives. So here we also make a report. We sort of filter out everything that's not clinically relevant. We have some caller-specific thresholds on the number of supporting reads. And in the end, we get a very sensitive fusion calling that finds sort of everything that we have been looking for, even though there are some cases that's close to the detection limit, but it still finds it. And another advantage here with the RNA compared to the DNA is that it's enough with one of the genes in the fusion to be included in the panel, and we can then find the other one. So we can find novel fusion partners. And here is an example of an image you get from Ariba showing you how the fusion looks, which can be nice to look at. Then we also have exon skipping, which is not called by the fusion callers. Here we use an in-house script that finds then exon skipping in the MET and EGFR. And here we also found all the tested exon skippings. To round this up, I just end with a few challenges with this project. So this was a national pipeline. So we here had to do this sort of coordination in national coordination. Luckily, we have had other bioinformaticians that has sort of been involved and been present at the meetings and been making lots of discussions. So this has been going quite well. Another thing, when it's a national project, you need to have the pipeline working at several sites. So a lot of effort has been done to make it portable. But this has also then increased the quality of the pipeline. So that's quite nice. And also when several people are running it on different sites, you find more errors and bugs. And so this has also been really nice for the quality of the pipeline. And finally, from the data's perspective, to find the artifacts and find good filtering is really a challenge. You want to get rid of as much of them as possible, but still retain all the true variants. So here there's been a lot of work done in these areas for all kinds of variants. And with that, I would like to round this off and open for questions to all of us presenters.
Thank you so much for very interesting presentations and for showing us really how much work it is that goes into developing a new test, and especially such a comprehensive test with detection of many important variants and markers. Really strong effort there. So yeah. We have now time for questions to Johan, Gilbert, and Jonas.